Thank you very much for the kind introduction and for giving me the opportunity to speak here today. So first of all, to confirm, do you see my screen and do you hear me?
Yes, perfectly.
Okay, then I will just go and give my talk. Also, some apologies, I'm a little bit sick, but the good thing is that it's an online conference, so I cannot give you my terms. Yeah, so first of all, I would like to introduce you to my research lines. So it's all centered about the technique that is called diamond magnetometry. And this is a technique that and why is that nice? Because optical signals can be read out much more sensitively than what you can normally do with magnetic resonance signals. And for that reason, the technique is very sensitive for a magnetic resonance technique. And we use now that sensitivity in two different kinds of ways. So on the one hand, we use the sensitivity to gain spatial resolution, so to measure things that are very small. And on the other hand, we use that happen fast. And today I will mostly talk about the one on the left, so to measure small things. So first of all, what is the challenge that we are interested in? So my group is concerned with measuring the detection of free radicals. And why would you want to do that? Well, they are very important in all kinds of biological processes. Like, for instance, cells use them to communicate with each other. They use them to trigger certain processes in maturation. But they are also part of the process, how we degrade food or how we age. But they are also involved in a lot of diseases, and this includes the one that are responsible for the most deaths worldwide, like, for example, in cancer or cardiovascular diseases. In bacterial or viral infection, you always see that there is some change in the free radical load of cells. And the reason is that they are an indicator of stress. So usually when there is something wrong in the cell, one of the things that happens is that there is a misbalance in the radical load. And while these radicals are very interesting, they are also very difficult for the state of the art because they are short-lived and they are very reactive, and there is currently no specific probes available for them. And with that, I mean, if you, for example, want to see a protein and where it is, you just get an antibody for that protein and then you can visualize it. But that is not so easy when it comes to free radicals because they are too small to make good antibodies. And then there are a few techniques that can tell you something about radicals, but none of them can really differentiate and map free radicals in real time. And the biggest problem is in getting spatial resolution. So there's really nothing that can allow you to map radicals at the nanoscale, and that is what we would like to do. So here a slide that I've taken from the literature, from somebody that works with a similar technique. And I told you before that these systems are very sensitive. And now here you have a chart. You can see here on top the magnetic sensitivity, so the smallest magnetic field that you can measure. And you want to be on the left here. And on the y-axis, you see the spatial resolution that you can get. So you want to be on the bottom left here. And so now there are a few techniques here that are very sensitive, like squids, for instance, and MEMS. But the problem is that my samples are live and they are at room temperature and they are in liquid. So most of these techniques just don't work. So for squids, you need superconducting, very low temperature to achieve this squid effect. And these sensitivities that are mentioned here is if you can bring the squid that has to be at low temperature very somewhere on your head, very far away from what they're actually sensing. And then the sensitivity goes down again. MEMS, for instance, that was here, that referred to like very floppy cantilevers that are used in high vacuum and low temperature as well. So that is also not compatible with cells. And then the Hall sensors here, those are typically like wires that you connect with your samples. And that's also not something that you want to do with biological systems. So what's really left is clinical MRI, and that would be to the top right on this graph here, so it's not even on the chart, and the work that we do with diamond. So what do we do exactly? So we make use of diamonds that contain so-called NV centers. So this is a defect in diamond that changes its optical properties based on the magnetic surrounding. And we take these defects that are in nanodiamonds and we bring them, as shown here, we bring them in a living cell. And then, we do all kinds of spin manipulation and we read out the signal that we see from this diamond here. And the diamond we use to measure the free radicals that are in the surrounding. So if you look at the picture here, so this is a picture taken on our microscope, and the blue blobs here, those are the cells, and the white particles, those are the diamonds. And then you can get, you can do all kind of, in this case, a spectroscopical measurement from one of these diamonds at this specific position. Then, if you want to work with nanodiamonds in cells, the first thing that you have to achieve is you have to bring your diamond particles in your cell. And there are a lot of cell types that just take up particles. So this is true, for instance, for macrophages that are here on the top left, and also for the macrophages in the movie. So they just pick up particles. So in the particle on the left are the particles in red, and on the right in the movie, it's the white stuff that are the particles. And you can see that these cells, their purpose in biology is to get rid of particles, so they are very good at taking up particles. But there is also a lot of other cells that take up particles, and there's also quite some cells that don't take up particles, but then you can use surface chemistry to make them pick up the particles, or there's also methods to deal with the cells. But in this case, what I will show today, we don't need it. So this is on these kind of immune cells that you can see here. And here it's kind of interesting here on the bottom right. That was just a curious thing that happened during that video. By the way, it's fast forwarded. So what you see here is what happens in about 8 hours time. And there's one cell that goes and grabs a particle from really quite far away here. So what do we do technically? So we have this, what's called a diamond magnetometer. And by now we have about 6 of them in the lab. And it's basically a confocal microscope that has a few changes. So you need very sensitive detection, and we need to be able to do pulsing. And this is how it looks like, how one of our instruments looks like in the lab. And this is the one physics slide that I have of what exactly we do. So mostly we make use of a sequence that is called relaxometry. And what we do there is we shine with a green laser on the diamond particle. And if you do that, the electrons here on this NV center in this energy diagram, they go up. And then they go down again and you get some fluorescence. But there is something special here. So that's this MS plus and -1 state. And if something was in the plus and -1 state, it will be less bright because there is also this dark state here if you go via this state. So what's cool here is that you can look at the brightness and see whether you were in this 0 state or in this plus -1 state. And after we apply a laser pulse, we also see that all the arrows end in the 0 state. So we can initialize our NV center in the 0 state that is bright. And that's exactly what we do here on the right side. We apply laser pulse. We make the NV centers bright, and then we probe after a certain amount of time whether the NV center is still there. And if there is no radicals present, or in this case gadolinium, that is a contrast agent in MRI, then this takes a very long time. If we have radicals in the surrounding, or if there's gadolinium there, the time is reduced sharply, what we can see here in the red. And this difference in time here is called the T1 time, and is equivalent with what they call T1 in clinical MRI measurement. The difference here is that this is from a nanoscale voxel around the diamond particle. And here is maybe our first measurement where we have done this in a cell. So this is a macrophage, and we do a measurement before in the blue, and then we add some chemicals, in this case DMSO, that is toxic to the cell, and then we follow the stress response, and afterwards we can collect the yellow curve here. Okay, so we can kill cells and watch, but there is also some more interesting stuff that you can do with this. So I told you that we get these T1 values, but most people are not really interested in some T1 values, but they want to know the concentration of radicals. And to have that, we made kind of a calibration, and we use these compounds here that are called nono-8. And if you add these compounds here in an acidic environment, they will split off this molecule here, and they will generate nitric oxide. And nitric oxide is a radical, and you can make it in a somewhat controlled environment. And then we can measure T1, and we know the nitric oxide concentration, and then we can make a kind of calibration for what kind of radical concentration we get, what kind of T1. And then we can later on take the radical measurements that we did and convert the T1s to an estimation of the radical concentration. So now there are a lot of different projects that are ongoing in my group where we make use of this. I would just like to show you a few. For instance, we are using this technique to measure the impact that certain drugs have on a cell, or to test whether a certain drug is working. Like for instance, a cancer drug or an antibacterial drug, they are causing a stress response by the cells. So we can add the drug and see what the stress response is. How they differ in their radical metabolism. And we found very interestingly that certain mutants that have more longevity, that they are behaving different in terms of their radical response. Then there is the immune response that I will show you some data of later on. And we also have a first clinical trial that we are running at the moment, where we are looking at the fertility of sperm cells, and where we are trying to correlate the radical response of the sperm cells with the clinical case, with the fertility of the couple. We have a project on viral infections, also a project on bacterial infections, but you can also imagine that you can do all kinds of non-biological things, like for instance, looking at polymer degradation or reaction intermediates. So now let me show you first data. So in this paper here that was published last year in Science Advances, we were looking at macrophages, and we made two types of nanodiamonds. The first type of nanodiamond was bare, and in this case, we know that the particles go in the cytosol, and we had a second type of particle that are coated with antibodies for the mitochondria. So we can target these types of particles to go near the mitochondria. And now if you look at our T1 data, again, low T1 means high radical concentration. Then what you can see is that, first of all, the particles that are in mitochondria, they have a higher radical load, which is what you would expect. And then we can add chemicals. And in this case, we added a decoupler. So this is a chemical that is turning off some reaction at the mitochondria. And then if you have the particle at a different location, of course, nothing happens. But at the mitochondria, you see a response to this trigger. And then here's a boring slide with all the controls where nothing happens. But let me use this slide to explain what we do. So we have here, on this case, the relaxation time, the T1 that I explained before. And on this scale, we have the concentration in radicals that we got via this calibration that I've shown you before. And then we have here gray lines and colored bars. So the gray lines are individual experiments from single cells, while the colored bars represent the averages that you can see here. We had particles and we added different chemicals, but we did not add cells. So if you don't add the cells, then you don't see a response by the cells. So we see more or less no differences here. The same happens if you have a cell, but don't add the chemical. So this is the ones that you see below. And in this case, we pretty much also see no response. Now, I would like to show you some single organelle measurements. So particles are in red, and you can see here that they are nicely together. And in this case, you can see here now the differences look different than before in the control. So here we again have the similar kind of measurements. And in this case, so for this compound, you can see that there is a concentration dependence, which is also what we expect from the literature. And here we have another compound that is, also I believe an inhibitor in this case. So we see a radical formation that goes down. And in this case, we are inducing some stress and we can see the radical formation going up. We can also do the same thing in a cellular context. So this is the mitochondria and the diamond particles when they are still in the living cell. And in that case, we see more or less the same response to these chemicals. While if we look at the particle at a different location, so this is at the mitochondria where we see the responses, and this is in the cytosol, where we see, first of all, a lot greater variability, but also no significant differences between these measurements here. Also to show you that you can also have this kind of data in a bit different way. So what we did here was we also have a macrophage. And in this case, we are measuring signaling with nitric oxide. And here we added a chemical that is called L-NAME, that is an inhibitor for this process. And you can see in blue here that if we inhibit the process, the radical formation is going down. So T1 is going up. And these controls here, so if we just add water or if we add a control that is at a different location, then we don't see a response here. And what's very nice here, so this time scale here is in minutes, so we get a lot of data points here. And then you can also have the data in this kind of fashion, so you can do some kind of imaging if you look at the trajectory of the particle. So in this case, we looked at a particle that goes to different locations, and then we map the magnetic field at the different positions. So with this, I'm already at the summary of my talk. So we have achieved uptake into cells. We have control measurements with chemicals that allow us to calibrate and to interpret different concentrations. And we have the first magnetometer. And we can do measurements that are organelle specific. So we can go to a specific location like the mitochondria and then do a measurement specifically there, which is something that of institutions that gave me money. And this is my research group, except for the small ones, those are my children. And I would like to thank all these people and of course, all of you for your attention. And finally, I would like to invite you to ask me some questions.